इस मॉड्यूल में हम बात करेंगे रिएजेंट्स और सप्लाईज की जिनकी हमें ज़रूरत पेश आती है फॉर डीएनए सीक्वेंसिंग यूजिंग टैक एज अ डीएनए पॉलीमरेज. Different buffers and solutions are required which should be prepared before time and should be ready to be used for the sequencing process. We need deionized water, ice cold. Then stock solutions of nucleotides are required. So keep in mind we need two different types of nucleotide triphosphates, one normal DNTPs and the second one DDNTPs, dideoxynucleotide triphosphates. And formamide loading buffer is also required for this purpose. Labeling mixture and DDNTP chain extension termination mixture is required. 10x of this buffer is required. And when we sequence the double-stranded DNA, sometimes there is a high degree of secondary structure. In that case, it is advisable that instead of using DGTP, we should use an analog which is 7-DESA 2'-DGTP. It is going to be helpful for sequencing of those DNA molecules that have high degree of secondary structure. Then we need 5x reaction buffer, tris chloride solutions, magnesium chloride solution, and the enzyme, the Taq polymerase or any other temperature stable or temperature thermostable DNA polymerase. But keep in mind when we use any other Taq polymerase, the requirement of buffer, the concentration of different solutions may have to be adjusted. Then Taq dilution buffer is required. That includes tris and EDTA at pH 8.8 and pH 8. Tween 20 is required, 0.15% non-det P40 is required to prepare these buffers. Then comes the nucleic acids and oligonucleotides. Oligonucleotide primer is required because we are basically working to synthesize a new strand of the DNA using the template DNA. So we need to have a primer which is oligonucleotide primer. We use that primer at a concentration of 10 picomolar per microliter, and if we look at the quantity of the primer, that is 6.6 nanogram per microliter, and this should be prepared in TE buffer. Besides gene specific primer, there is an option that we can use universal primers. A list of universal primer is available which can be purchased from the commercial sources. Then we need the template DNA, which is normally double-stranded, denatured plasmid DNA, and we convert it into single-stranded DNA by denaturation process. We need single-stranded DNA, 500 nanogram or 1 microgram of denatured double-stranded DNA. This method is applicable for both the types of DNA molecules, double-stranded DNA or single-stranded DNA. So the starting concentration can be different. For single-stranded DNA, we use 500 nanogram. Otherwise, we use 1 microgram of the double-stranded of the DNA. And this much amount is required for 4 different reactions, which are labeled as C reaction, G reaction, T reaction, and A reaction. Then we need radio-labeled nucleotide because we want to fix one end of the newly synthesized DNA strand. So we may have different variants of DATP which may have P32, P33 or sulfur 35, or another option could be P32 labeled oligonucleotide primers that will help us to label the end of newly synthesized strand. Special equipment, microfuge tubes are required, and if we are working with the microfuge plates, then 9612 plates can replace the use of microfuge tubes. Programmable thermal cycler is required. Water bath preheated at 65 degrees centigrade for incubation purpose. This table shows you the 10x labeling mixture. Now, what is the concentration of any particular ingredient or reagent in the reaction mixture can be seen here. That we need for labeling 1.5 microliter of DCTP. We look on different columns, like this one is the labeling that what kind of DD NTP we need, that is DDCTP, DDTTP, or DDATP, or DDGTP in different tubes. This explains what volume of DCTP, normal DCTP, is required, and besides these nucleotide, what else we should use, in what volume we should use it, so that the end volume of water can be decided and the solution should end up with the equal volume. So by this table gives us an idea how to prepare the solutions to proceed further for sequencing.